I guess can just start. So thank you everyone for coming today. And and if you could introduce yourselves, I guess we can start from Miss Lorraine at the top.
Okay. I am Lorraine Doucette. I run at Arundel Community College. I run the medical lab technician program. I also run the phlebotomy program and trying to start up the medical lab assistant program. But as of June, I'll be going back to my old haunts, which was the University of Maryland Department of Medical and Research Technology at the School of Medicine. So I'm going to be leaving Anne Arundel to go teach the 4-year students in lab medicine. So my background is as a lab person. I've been doing lab work for all my career.
Nice. And then Dr. Nylish.
Yeah, thanks, Ryan. So I am Dr. Nilesh Kalyanaraman. I'm the health officer for the Anne Arundel County Department of Health. I am an internist by training, and I've been in community health for 13 years prior to taking this position about 7 or 8 months ago now. So excited to be here and looking forward to the conversation.
And then Dr. Ray.
Thanks, Ryan, for organizing this and inviting me. I'm Stuart Ray. I'm a professor of medicine and infectious diseases at Johns Hopkins. I've been at Johns Hopkins for about 30 years. Came as an intern, and I guess you could say I never graduated. I was born in Baltimore and grew up in Nashville, Tennessee, but I've been around for a long time, and I am vice chair of medicine for data integrity and analytics at Johns Hopkins, and have some laboratory activity in studying viral evolution.
And then Dr. Annie Antar.
So hi, my name is Annie Antar. I did an MD and a PhD in a combined program at Vanderbilt in Nashville, and that's very close to where I'm from. I'm from Northern Alabama. And then I came to Johns Hopkins to do my internal medicine residency and then stayed for a fellowship. And Stuart Ray has been, Dr. Ray has been a mentor of mine. And now I focus on HIV, but have been on the wards. Both Dr. Ray and I have been on the wards taking care of COVID patients, and we're both heavily involved in research, both basic science and clinical for COVID at Johns Hopkins. So we're happy to be here, and thank you so much, Ryan, for inviting us all.
No problem. Okay, so since we didn't, I'm Ryan. I'm a first year at Arundel Community College, and I'm looking for, my major is environmental science. And to start, what are some precautions that you guys could suggest for people to lower their chances of getting COVID-19, or who to contact, or what you could do if you start to notice symptoms? Like just like a basic guideline for people. I guess, does everyone has any thoughts?
I would say wash your hands. Just wash your hands with soap and water lots and lots and lots of times. Just wash your hands.
There's a few things we recommend everybody do. So washing your hands frequently with soap and water, definitely one of them. If you don't have access to that, hand sanitizer is fine, but we actually prefer soap and water. It's actually better for a number of reasons. Making sure that you're disinfecting frequently touched objects, particularly those non-porous metal, plastic. They can harbor virus for a longer time. Stay at home, please. Stay at home. If you don't have to go out, don't minimize the times that you are going out. So if you go to the grocery store a few times a week, try to get as much as you can in one shot, so you're not going out as frequently. Wear masks. The masks are something that we've learned about as in the process of going through COVID and learning about it. For those of us who are working in this field, it's really interesting and fascinating to learn with the public in near real time what to do. So the masks are key. Make sure that you're keeping 6 feet from others, so social distancing as much as you can as well. Those are kind of the key things that are going to help. And then particularly if you're ill, staying at home or where you are while you're seeking advice is really important as well. The more we don't expose others to our symptoms, the better off we'll all be.
Does anyone else have any more thoughts on the question? Okay, cool. So the next thing, before we answer any questions from the audience, is there anything that you guys specifically want to talk about? If you guys have any slides or anything? Maybe we can start from Professor Lorraine.
Well, I put together like a little PowerPoint. It kind of goes into what the COVID and the coronavirus is all about. So I can share the screen with that for that and kind of go through that if you'd like. Probably take about 5 minutes or so to kind of go through, because it also goes through PCR, and there was a couple questions about what's an antibody, what's an antigen, and I also got information about how the antibody tests, where are they getting how are they testing for those antibodies? So look that up too. So, oops, let me go share screen, and let me share the screen. Can I share the screen? I don't know if I can share the screen. Huh. Host, disable, oh, you've disabled the participant screen sharing. So I can't share the screen. So Ryan, can you hear me?
Yeah, I think he's looking for
Yes, yes,
Oh,
sorry.
okay.
I was muted. I should be able to fix it.
Okay.
Just give me 1 minute. While I fix that,
Yeah.
Yeah.
I can make a couple of comments for a little context maybe while you're getting that up. And I'll stop when Lorraine has her screen shared. But we know from experience that there have been respiratory coronaviruses for many years. And there are some with catchy names like OC43 and NL63 and others that cause the common cold. From those, we've learned that people can get them more than once in a lifetime, unlike some other viruses, so that people can get OC43 and then months to years later, they can get it again. We don't know a lot about that because it hasn't been a major focus of research, but we do get a sense that resistance to those viruses can wane, perhaps because they're so mild, we don't know. So there's a lot that may not translate to this new coronavirus. We know that there are many coronaviruses out there, and one of them transferred into humans in 2002 and caused an outbreak called SARS, severe acute respiratory syndrome, and that caused a great deal of concern because it spread so rapidly. Fortunately, people did not spread that virus when they were not symptomatic, from what we can tell. And so there were no transmissions from asymptomatic people to other people during that outbreak. And that outbreak ended pretty quickly. Subsequently, there was an outbreak of another virus that was subsequently called Middle East Respiratory Syndrome, or MERS, that also was caused by a coronavirus, very distantly related to the one that caused SARS. And that one was not spread very efficiently among people, and that outbreak was contained as well. And that brings us up to the recent present, when in November, December, people started getting sick in the Wuhan region, and that prompted this awareness of the epidemic, which was announced December 31, 2019. And this epidemic has been caused by a beta coronavirus, and we'll talk more about that, but I think Lorraine's ready to present her slides.
Yeah. Let's see. So let me share the screen. Still saying disable participant screen sharing. Let's see. I don't think you guys can see that.
Not yet.
Yeah.
So it's worth at least mentioning.
Oh, wait, here you go.
You got it?
All right, finally. All right, share.
Zoom conferences or
Yes.
conferences. There we go. Great.
Here we go. Here we go. So, okay, so this is the virus and a little cartoon version of it. And it's called the coronavirus because it looks like it has a crown around it. So there's many different types, as Dr. Ray was saying. So there's different, here's the spike protein here. And so there's three different main proteins here, the E protein, the S protein, and the M protein, and they do different things dealing with this particular virus. So, and this is exactly what he was saying about the common colds. So about 20% are caused by coronavirus. And then we've got some others that are out there too. So about 30% of colds are caused by viruses that we don't even know what causes them. You're just told, well, you have a cold. So we don't really have we don't know the virus. And then there are the coronaviruses of the class. So this is exactly what we'd already covered. And then this is the latest one, the SARS-CoV-2. So a lot of people are confused as to how to actually say what is what. SARS-CoV-2 is the actual virus, and then COVID-19 is the disease. So a lot of people are caused by this particular virus. So so that makes it a little easier to kind of keep in mind.
If I could just clarify that, if you go
Sure.
back one slide. So COVID is coronavirus infectious disease. So that's COVID is the name for the disease. SARS-CoV-2, because it's coronavirus 2, the it isn't part of the virus name. So
Oh, okay.
SARS-CoV-2 is the
CoV-2.
name of the virus.
Okay. So this, okay. So sorry, I got that one part wrong. So then they found, the researchers in Texas, Austin, found that the S protein, the spike protein, binds to the angiotensin converting enzyme. And this is kind of interesting how this works, because it binds here, and then that's kind of like the key, and it binds onto the pneumocytes, which are the cells in your lungs. And then when it does that, it unlocks it. So, and then it goes straight into the, it's like a lock and a key mechanism going from this virus straight into your cell in your lung. Very, very, very specific. So there's been some research going on about this lock and key mechanism. I'm sure, one of you could talk about this a little bit about, how this could lead to treatments or vaccine or whatever, once we get this figured all out. And then how does it replicate? It's a single strand RNA, and it basically hijacks the messenger RNA in the cell nucleus, and then it starts making copies within that cell's nucleus of the cells, hijacking the DNA of the cell to make its own proteins. And then once it fills up with all the viral particles, it kind of just destroys that cell. And so there's two methods that we can use to diagnose the COVID or the viral infection. I'll just say, leave it at that. So there's one is polymerase chain, it's PCR. So first, specimens collected by nasopharyngeal swab in a patient who's got symptoms of the virus and a doctor's order. So these are what you see all around, these collection sites. They keep calling them testing sites. We've got a new testing site open. There's no testing going on in any of these sites. All they're doing is you're driving up, somebody is taking a very long nasal swab, sticking it way back up, so far back up you think it's touching your eyeballs, because it's way in the back of your throat, way, way, way back there, and then putting it into a special tube that has a special fluid in it. So one of the problems about getting people tested for this was that we ran out of the swabs. There was a lag on how many swabs we had and the liquid media. We had to actually manufacture our own in the lab. The people were making their own media instead of having it coming from a manufacturer. So once that bottlenecks have been taken care of, then there's no shortage now of the nasopharyngeal swabs. And so what happens is that the PCR polymerase chain reaction is going to amplify the RNA, and then what it does is it just keeps making up little blocks of it, and just keeps making it over and over and over again. What is typically done is something where it's 35 cycles. So there's 3 stages: denaturization, annealing, and extension. This takes time. So you're actually going from a double-stranded DNA going into 2 pieces of single-stranded. So just think about it like a zipper that you've got closed, and now you're unzipping the zipper. So you have two sections. Now you're going to make copies of each side of your zipper, and you're going to make multiple copies. Well, that takes time. And so then what we're doing in the lab for the coronavirus is that we're using reverse transcriptase PCR. It's a slightly different technique, and it's where you're making this complementary DNA from the RNA, and then you're amplifying that. But again, it takes time. It's at least 3 hours, can be as long as 6 hours. But we've tagged it. So this is an example of two different patients, for example. What happens is that once you pass this threshold level, each time it's going through the cycle, it's being amplified, and it's going up and up and up and up and up. And as it's going up, there's a fluorescent dye attached. So we can watch in the lab, we can see the signal going up and up and up and up and up. And this one has 1 million copies. So it rose much faster than this one, which only has 4,000 copies. So that's how in the lab we use this instrument that could take 3 to 6 hours till we see enough of the fluorescent signal to then say, okay, this is not only is it positive, but this is like how much of a dose of it you have, I suppose you could say. So you have plenty of this particular sample because it copied itself so fast. So the second method is antibody testing. So this is some basics about antibodies and antigens. An antigen is anything foreign. You can have ragweed, you could have allergies, all of the dust and the grass, those are allergens. We sniff them in and our bodies make antibodies against them. Or if you're allergic to poison ivy, you touch poison ivy, now you have a rash in your skin, you've got antibodies to poison ivy. So it could be something simple, but it could also be against pathogenic things like measles and mumps or HIV or hepatitis. So you make 2 different kinds of antibodies, an IgG and an IgM. IgM is made first and then followed by IgG. And the new method that they have out there, it's an instrument called the Architect, that actually measures IgG, and it's actually manufactured from, it's a mouse monoclonal antibody. So it's an antibody made from mice that is then coupled with a specific marker so that we can check for the antibody in the patient's sample. So that particular method is a pretty accurate method, but there's lots of ones out there that are not very accurate. And then, well, if you had to pick a gold standard for which one's more accurate, you'd go for PCR. But you're going from an antibody test you could do with maybe 10, 15 minutes versus 3 hours. So, and then what I'm advocating for is, and one of the other reasons why it's a long time, is that there's a lack of laboratory professionals. We're getting, there's a shortage, a huge shortage. So, and then why does it take long to get the lab test results? And then a result's only accurate if the sample's collected correctly. So a lot of times in the lab, what I'm hearing from my lab friends is that because of how stressed everybody is, and they're just working super long hours and stuff, what happens at these collection sites, we're getting samples that don't have labels on them. They're just an unknown nasopharyngeal swab. Well, that is meaningless, but it takes up everybody's time. Unlabeled blood tubes or clotted blood tubes, which is another big problem now that we're seeing with the virus. It tends to, in some patients, make their blood clot. And then the sample's actually still up at the nurse's station and never made it to the lab. So we can't test something that we never even got. So, and then the huge personnel shortage. So there's 9,000 techs needed a year, but only 4,000 graduate. So that's a huge, huge, huge issue. And so that's, hopefully there's people out there now kind of understand that the lab profession is, we're professionals and we're truly doing a huge, huge job for getting all this kind of testing done. And in the meantime, we're still doing testing for people who have heart attacks and all the other kind of testing that's routinely kind of being done for the, say, the patients who have other things wrong with them. And so one of the things that people don't realize is that without the clinical lab, we'd be practicing medicine like the 1940s. If there was no modern laboratory and all, we would have to be back to the 40s or the early 50s. And yes, we do go to school. We can hear this a lot. People will actually come into the lab and say, do you go to school for this? It's like, yes, we go to school for this. We have associate degrees, bachelor's degrees, master's degrees, and we work. This is one of the things they keep talking about, machines in these in the either reporters or whatever. They keep saying, well, the people in the lab have working on machines. No, a car is a machine. We work on incredibly complex instruments. So a little plug there for the right terminology. And then, so that's my references. So, but that's kind of a quick little overview of the lab profession, my little plug of like, hey, if you're interested in the lab field, do like Dr. Ray, go into the lab and never come back out. So you make your career with the lab. Because it's a rewarding profession.
And last week was Medical Lab Professionals Week.
Yes,
so thank you
was.
for what you've done, Lorraine.
Well, thank you. Yes, yes. So yeah, people didn't even have a chance to stop. They were, and I have friends who work at Hopkins in the virology lab, and they're working 24/7 on their method. They're producing thousands of tests a week, and they're dressed in basically those suits that you wear like in the movie Outbreak. So it's hot, it's grueling, but the virologists wouldn't have it any other way.
Yeah, big shout out to Dr. Hiba Mustafa and Dr. Karen Carroll of Johns Hopkins. They've been leading the efforts with our virology lab, and there's several different platforms that they're using, several different instruments that they're using to do this testing, and they've done a wonderful job.
Right, right.
And thank you, Lorraine, for all the work that you've done.
Sure. It's like just saying, oh, well, you just tested on a thermocycler. It's like, well, that doesn't sound very exciting. But so it does, it's a workhorse. It's like, yeah, you heat this stuff up, and PCR has been around for at least 15 years now, at least, maybe longer. When it first started, I mean, you actually make all the materials yourself, and you had maybe one or two pieces of genome, and then that was it. And then you were going from there. It was like the Wild West. Now they've got things a little bit narrowed down some, so that's good. But then every once in a while we get a weird disease or like COVID or like, the coronavirus.
So that was it for presentations, and now we'll start answering questions from the audience. So I have some pre-written questions, and I'm getting some questions live. To start, this was a big question, was what would a hypothetical scenario of everything suddenly opening back up look like, and what would the effects of it? Could Dr. Nylish and Dr. Ray maybe answer that?
Sure. So the hypothetical scenario of everything opening back up is very bad. All the efforts that we've been taking over the past 6 or so weeks at this point, the distancing, the infection control, what it does is it slows down the rate of spread. One of the things we talk about is how infective, if you've heard the term R naught, it's just a fancy way of saying how many people do you spread it to? And if you spread it to more than one person, then it keeps going. And if you're able, and if we can get it to where we spread it to less than one person, then it slowly can die out. So all our efforts have been to slow this spread. Our big challenge is how long can we go this way? We have to, at some point, open things back up again, but we should understand that as we do that, we'll start to see spread increase again. And so that's where we talk about what are the things that are needed to be in place in order to start to think about opening things up again. And that's where the testing comes into place. And we're really focused on not just getting as many people tested, but testing every single person who has symptoms the day that they start having symptoms. That's really the target that we have in terms of testing. And then along with that comes contact tracing, which is a term that is now in the popular parlance, but has been something that public health has been doing for decades. And it's just a term that means if somebody has a communicable disease, we want to know who they've been in contact with, how did they potentially get it, and then also who did they potentially spread it to. And then we do isolation and quarantine. So for somebody with the symptoms, we ask them to self-isolate, and if they can't, we will help them with hotel stay or whatever supports they need to be successful so that they don't spread it to others. And then for the people that they've come into contact with who may have been exposed to it, quarantine. Quarantine looks the same as isolation, but it means you don't have symptoms, but you may develop them. And so the same thing, making sure that they can quarantine, providing supports if they can't. That really needs to be in place where we can do contact tracing for every single person who's positive so that we can limit the spread as much as possible. Those are kind of the two key pieces. And then the other pieces are making sure there's enough PPE so that we can gowns, gloves, face masks, face shields, so that we can actually do this testing, so that we can actually provide care. And then a lot of our focus has been on hospital capacity as well. Because the more we can slow this, the better we can provide care to people who are getting sick and who need that kind of care.
I have a question for you, Dr. Nilesh. Is Maryland doing contact tracing right now? Yeah,
so we're doing contact tracing. Different counties have a slightly different approach. Here in Anne Arundel County, we are contact tracing every single person. Some counties have focused on more vulnerable populations. It just depends on their volume. But what we're gearing towards right now is making sure that we have, have in place a system where everybody can get contact traced. So we're actually right in the middle of working out what that looks like in terms of county, state partnerships. But the core of it is that we want to make sure that we have 99.9% fidelity to this. And so that means being able to do contact tracing and supports for those who are vulnerable or don't have as many resources, and then having a shared resource at the state level as well, so that we can do large volume too. So it's that both and kind of scenario.
One thing that analogy I've heard that seems useful is that this flattening the curve is a little bit like wearing a parachute on your way down from an airplane. And the question is, when can you take off the parachute? A lot of people would say that it's a good idea to wait until you are on the ground safely.
Yeah,
some people are going to want to take the parachute off early. And so I think it's worth describing what the splat looks like. We've seen some of that splat happen. We've seen plenty of surprisingly young people who do not have premorbid disease die or get very sick with this disease. And so I think we shouldn't pretend that it doesn't make young, healthy people sick. One of our colleagues' brother, 50 years old, died from COVID, previously healthy. We have, he was not a healthcare worker, but this happens. And we don't understand why some people get sick. We know from Italy that some surprisingly young healthcare workers got very sick and died from COVID, making us wonder whether the amount of inoculum or some other thing, maybe fatigue, but we don't know what it was, but some young people got surprisingly ill. And I think the experience in Italy also tells us something, because in Italy, they were so overwhelmed in their hospitals and their ICUs that people were not getting standard care. And that's something we tried very hard in Maryland to avoid. And I think we did a pretty good job. We went really to the limit on some of our hospitals in the region that filled up their ICUs, filled up their capacity for taking care of people with COVID, had to transfer patients between hospitals to avoid being overwhelmed. If we had crossed that line to overwhelmed, then people with a stroke or a heart attack would have been turned away. And that happened in some of these other places. So we have to realize that we've dodged the bullet. We sort of ran close to the edge of the canyon and didn't fall in, but we're still walking at the edge of that canyon. And as we think about what we do to try to revitalize the economy, because that's clearly a huge impact for many people, we have to realize that overwhelming our health system would be devastating.
Yes, absolutely. We all have to remember there's no proven treatment and there's no vaccine, and we don't know when those are going to come online. And our only defense against this virus is strong public health measures.
Right. It's also keeping people who potentially would be visiting a hospital away from visiting a hospital because they're trying to deal with it going to clinics and things, because they're afraid that, oh my gosh, I'm going to go into that ER and then that's it. I can't have any visitors. I can't have any of this, and basically they are kind of on their own. Everything's kind of like on lockdown. So people are self-selecting away, which can be kind of dangerous. So, that's also is a health issue too. So we want to make sure that people actually do go in for medical care when they need medical care and don't try to, just do the minute clinic or any kind of like just clinics and stuff. That's not what they are designed for. But that can be quite challenging.
Yeah, Lorraine's right about our EMS folks are seeing a lot more severe heart attack, stroke kind of patients than they normally would. They're now usually they transfer people with mild symptoms and some people with severe. Now it's a lot of people who haven't sought treatment because of all the things we're saying, right?
Right.
We see these costs build up elsewhere as well in terms of lives.
Yeah, I mean, they're not COVID positive, but they just don't want to even go there because of all the lockdown stuff. And the not having any visitors policy is just, because of COVID, that I totally understand that, but it's also then keeping people with other medical conditions away. And then they're waiting until they can't take it anymore and it might be too late.
Yeah, that is a tough policy, the no visitor policy at the hospitals. I took care of, COVID patients on a COVID ward for a week last month and going back next week. And that is really tough on patients. It's tough. This is a tough disease. People get sick and then they're lonely, and because they can't have visitors, and then it's hard to bring the things that they need, if they want their cell phone charger, for example. That was an issue, trying to get it in. That can be done, but it has to be coordinated. So it's tough. This is a tough experience for people who are sick with it in the hospital and at home.
And I think it's worth saying that if people wonder, like, I have or suspect I have COVID, when should I go to the hospital? Obviously, there are many ways you can get sick, but the real danger sign is breathing problems. So if you can't catch your breath, if you can't walk across the room without catching your breath, if you're short of breath at rest especially, those are signs that this is really dangerous. Many people do okay with COVID, and as long as it's just fever, just cough, then it's generally tolerable. But if you become short of breath, that's a time to seek medical care. You can do that but you need to seek care.
This is a good time to plug the Anne Arundel County Department of Health. We can answer your questions if you have specific questions. If you can't reach your primary care doctor for any reason, or you don't have one, we're available to both answer questions, and if it's indicated, do testing. Our number is 410-222-7256. And we're open 7 days a week. So from 7:30 in the morning till 9 at night, Monday through Friday, and then 10 to 4 on Saturday, Sunday.
There was a live question. There was a couple of live questions. And the first one is, how are HeLa, HeLa cells helping to find the vaccine? And could Dr. Annie answer that?
Sure. So that's a really great question. I just wanted to tell people what HeLa cells are. So these were cells that were isolated actually without the knowledge of an African-American woman named Henrietta Lacks, who subsequently died of cancer. So these were cancer transformed cells. They turned out to grow very well in the laboratory. And then the researcher at Johns Hopkins who isolated them has used them and then shared them with the scientific community, again, kind of without the knowledge and consent of the patient or her family. So this is quite a controversial thing to do. And this is the type of thing that cannot and will not be done these days, now that there's kind of ethical institutional research boards in place to prevent that kind of thing from happening. But HeLa cells have been important in science in numerous ways. Her cells have helped people understand basic mechanisms of disease, viral replication, many important things. So we're all very grateful for HeLa cells. And Johns Hopkins has a very nice remembrance once a year. It's a big symposium to remember Henrietta Lacks's contribution and to kind of make up for what happened. So HeLa cells, they're not the typical cells that we're actually using for replicating coronaviruses. They can be transformed with ACE2, the receptor for SARS-CoV-2. And in that sense, then they can support replication of the virus. But typically the cell line that is used now is Vero, which is an African green monkey kidney cell line. And then also kind of primary cells from human airway epithelium, I think are the main ones. But yeah, we're grateful for that, for the HeLa cell.
There was another question, and that was, there's conspiracy that COVID is a man-made virus, and is there any validity to this claim? And could Dr. Ray answer that for us?
Yeah, so it's the history of this virus is only incompletely known. We know that it's a beta coronavirus. There are alpha, beta, gamma, delta coronaviruses. This is a beta coronavirus. The most closely related virus to it that we know of was a virus isolated from a bat in Yunnan, about 1,000 miles west of Hubei, Wuhan, where this epidemic started. And so we know that virus was isolated in 2013, just in a survey of bats. We certainly have not sampled bats in a consistent way that we know that there's not a more related virus than some other bat. It's just that sporadically people do this. And actually that virus was isolated from the feces of a bat in Yunnan in a cave. So that's the most similar one. And so people have wondered whether they took that one and they just changed it a little to make it into this SARS-CoV-2. However, there are more than 1,000 genomic differences between that virus that's the most similar we ever knew of in Yunnan, 1,000 miles away from this bat that arose near Wuhan. It seems unlikely that one was used for the other. And so when we look at this virus, it has a number of features that make it unique among the close cousins that it has. It has an insertion of some basic residues, some arginines just before the junction between S1 and S2. S1 is the binding part of the spike protein that binds to the ACE2 receptor. And then the S2 protein, the second half of the spike protein, has the fusion domain that has to insert itself into the target cell membrane to fuse the virus with the cell. And that cleavage is an important part of the cycle, the lock and key mechanism that Lorraine talked about. So that these basic residues make the cleavage more efficient. And so people have wondered whether that could have been engineered. I think based on my 2 decades of virology work, I'm confident that we don't have any human virologists smart enough to have engineered this. More importantly, there would have been no way to test it without having huge numbers of people to test, because you can't test on one person, you'd have to test on many people. And that kind of experiment just is very hard to hide. Knowing all the different elements of this, I think it's exceedingly unlikely that this virus was engineered by a human. Now, what no one can say is whether someone might have isolated this virus and been growing it in a lab somewhere to characterize it, and then accidentally carried it out of the lab. And that has been one of the stories that's been told. It's all based on hearsay, and I think does not really contribute to our understanding of this virus much. So I would say that it's one of these things that you can get yourself embroiled in, but there's no truth to be known based on what's available right now. And I think the more important piece is that there's not any evidence that this has been engineered by a human, but if it was accidentally released after having been collected somewhere, that's a hard thing to say.
That's interesting.
Yeah.
Another question that has come up is how does COVID-19 compare up to the Black Plague in relation to how similar it is and how, I guess, the effects of it compares as well? And could Dr. Annie Antar answer that?
Sure, I'm happy to do that. So the Black Plague it's a bacterial disease caused by the species Yersinia. And there's a couple different types of it, bubonic, pneumonic plague. It's actually still endemic in animals in different parts of the world, including in the southwestern United States. The mortality of the Black Plague, if you got it, was anywhere between 50 to 100%. So a very high percentage of people who got it, and they typically would get it from animals or perhaps from respiratory droplets from humans. Most of those people would die without treatment. So nowadays, people don't die of plague for the most part because there's actually treatments. Our antibiotics treat it and treat it pretty well. So coronavirus is different. It's a viral disease. It's very easily spread from person to person, as can be seen in what happened in the last 3 months. The mortality rate is much lower than the 50 to 100%. So current estimates of the case fatality rate depend on the country that you're in, which is probably because of the difference in who's getting tested, but it's anywhere between 2 and 10%. The higher numbers are probably in areas where they're not doing a lot of testing. So there's a lot of symptomatic mild cases that are probably not being tested for. So it's the case fatality rate is under 10%. It's likely under 5%. And then more than that, it's just hard to give you information right now. So most people will not die with SARS-CoV. Most people, we think, are symptomatic, although there's recent reports indicating that that may not be the case. But the people who are symptomatic, there's a very large range of disease. So again, there's some percentage that's asymptomatic, and all the scientists and epidemiologists are trying to figure out what percentage that is. So some people never get symptoms. Some people get only very mild symptoms, cough, shortness of breath. A lot of people get moderate disease where they feel short of breath or coughing, they have fevers, they don't feel well, they're nauseous, they might lose their sense of smell or taste, might have some diarrhea, find that they can't eat. These are all very common symptoms. Have a headache, that's another symptom. And then some people in the second week of illness will progress to having very serious respiratory decline. And that's usually why people are going to the hospital. It's because they'll need oxygen to basically support their lungs. And some people will need to be mechanically ventilated. Some people will have some element of kidney failure or even liver failure. And then they need hospital and ICU care.
Dr. Antar used a term that I think is really important for people to understand well, case fatality rate. It's been a source of a lot of confusion because you see these numbers, and an important thing that people sometimes do is apply the case fatality rate to an estimate of the number of people infected to try to estimate population mortality. And you have to realize that the word case and the term case fatality rate is really important. So a case is a person who actually is recognized medically to have the disease. Infection mortality rate is another way of thinking about the other thing, which is what proportion of all people who get the infection die. That's very different from the case fatality rate, which is the fatality rate among people who are recognized as cases. So when you see big skewing in the numbers between different estimates, one of the things to realize is that case fatality rate is not the proportion of people who get infected who die. It's the proportion of people who get infected and get sick who die. It's an important distinction. And I think tying some of Lorraine's comments with this idea is the problem with testing, that if people are using tests with very variable test characteristics, and recently there was a report out of Santa Clara County in California, there have been some other reports with tests that are not well standardized and with sampling methods that are not well standardized, that you can get some very different estimates of the proportion of people who've been infected, because antibodies could be a marker of prior infection, but there could be, for some of the tests, some cross-reactivity. Yes, Lorraine?
Well, I was going to say that one of the things that I've been told about from the friends in the lab is that there's some of the antibody testing methods, and they have this huge list on the that come with the reagent, and it will say cross reactivity with blah, 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 all those viruses you had said, and and again the 30% of all colds are caused by a coronavirus. So you could be coming up positive on this antibody test when actually it's detecting the fact that you had a cold. So it's not saying you actually have COVID. It's saying, we're detecting your antibody to the cold, and there was a question earlier about, does antibodies mean immunity? Not necessarily, because you can have antibodies to the flu and then get the flu again, or we don't know. This virus is too new to know. I mean, we have to remember this is only April. It's been only here in the United States pretty much like ramped up since Febrough. That's 3 months. I mean, 3 months. And so none of the methods for the antibodies are FDA approved. They've been just thrown out there. If they were FDA approved, they'd still be sitting in the FDA labs being methodically tested. It takes usually about 2 years to get a new method to actually come into the lab, and instead the techs are the ones doing all of the linearity checks and all of the validation studies and stuff, and they're finding that there's so many cross reactivities because they're not specific enough, or sensitive enough. So we have a lot of false positives and a lot of false negatives. So that the PCR is a gold standard. You see that bit of the molecular DNA or RNA, boom, you've got the virus. So that one we know, but the antibody, like, well, could be this, could be that, so it's until they've got those little cartridges that look like pregnancy tests, that like you do finger stick and stuff, those are just probably not worth even buying the kit, because it's just going to give false sense. Because when you have those kinds of things, you can't really validate each individual one.
Another popular question was, even though the rate of deaths for people were mostly for people who are 60 or higher, are the severity of the symptoms same for all ages, or how does it differ? Could Dr. Lorraine answer that?
Me? You said?
Or
Oh.
yes.
Okay. The ages, it most of what we're seeing, and please chime in, the MDs on this one too. What I'm seeing, what I'm hearing about is that it's mostly the comorbidity stuff. So if somebody is into their 60s, their chances of having high blood pressure is much higher than somebody in their 20s, or they might have diabetes, and type 2 diabetes, which they developed in their 40s. So those two things together, those tend to be high risk factors for having a worse outcome, it seemed like, from what I was reading about. People in their 20s though could still get the disease, and there's a lot of people, a lot of men, and it tends to kill more men than anybody else. It tends to sicken more men and kill more men than women. But it could also be that men in their 40s, they're not getting medical care because they don't go get medical care. The average woman goes to medical care every year. So it could well be that these men have morbidity things, and they just don't even know about it. But, you know, for the 20 to 30 to 40-year-old, yeah, that is a problem that, you know, they need to take it seriously. There's a big myth that if you're young and healthy that you won't get this virus. And that's not necessarily true.
Well, I think another thing to emphasize is while the person might not get sick from the virus, they may spread the virus. So
Right.
If you're a young person thinking I'm going to be able to dodge this thing, I don't think many young people would really want to infect family and friends and other loved ones or be responsible for a big outbreak. So even if
Right.
someone's not as susceptible to disease because of their age, they may well be more efficient as spreaders of the disease.
Or they may be undetectable. They may actually have the disease, but, you know, we'll tell that from better antibody methods later on, once once the crunch stops, then we can actually start measuring the antibody levels in these totally asymptomatic people. So maybe there's a lot there's a lot of infected people, but they were asymptomatic and they're young and otherwise healthy, and they actually got the virus. We don't know. We have no idea.
I just want to caution that people in every age group have died of COVID-19.
Yes.
And the strongest risk factor for disease is age, but there are others too, just like you're talking about comorbidities. I was just talking to Dr. Eileen Scully today. She's a researcher at Johns Hopkins interested in sex differences in multiple viral diseases. She said, looking at the data from New York City, it appeared that men were more likely to be hospitalized than women, but once they got to the hospital, so once you have,
Okay, once you,
then they had this, they appeared to have this.
that's good. That's good.
But there's a lot to be learned there.
Right.
And the other thing I want to note is that there is a disproportionate impact on different groups in terms of racial and ethnic
Yes.
background, and we're seeing that. We're seeing that in our county. We're seeing that in our state. We're seeing that there's a greater proportion of African Americans and Hispanic who are getting this disease and who are suffering mortality differences that are higher than other groups, and that has to do with a number of factors, both historical issues around economic access, around access to healthcare, around racism. And we're seeing that in our communities. So what we're seeing here in Anne Arundel is that we're seeing disproportionately, probably about twice the number we would expect, or percentage we would expect, in terms of African Americans being positive for COVID amongst our positives in the county. And so a lot of the issues we have, particularly around poverty there, that people who are poorer, I'm not suggesting that race and ethnicity are the same as poverty, but we do know that there's a disproportionate number of people in poverty who are, or have lower wage jobs, are in racial and ethnic minorities, don't have the options we have to shelter, to stay at home, to do telework if that's possible, to be able to isolate or quarantine in the same way if you have multiple rooms. And so they have to go to work, take public transportation. And so there's just a lot of those factors that pile up to also have that differential in terms of who's getting it. And so a lot of the talk about reopening, I would suggest that it has a disproportionate impact even where we are now, but when we open, it will hit those communities even harder.
Another question was, so one person said my sister is immunocompromised. What steps should the family take to, I guess, protect the person who's immunocompromised? And could Dr. Nylish answer that? And could Dr. Ray answer about, can you explain about what immunocompromised means?
Why don't I let Dr. Ray start, since it's probably better to answer in that direction.
Okay.
So yeah, immunocompromise is a complex topic. I think it's worth saying that the immune system has many components. As Lorraine said, there's antibodies, there's also, and the antibodies are made by B cells. There's also T cells, which play a different role. And then we have a big component of the immune system that's called innate, meaning that the ways that it recognizes danger are wired into our genome, doesn't have to be adapted, doesn't have to be trained. So these aren't the parts that respond so much to vaccines as are sort of hardwired into our DNA. When we talk about immunocompromise, mostly what we're talking about is things like that impair the adaptive response, the B and T cells. So when somebody's received an organ transplant and we try to suppress their immune system so they don't reject the transplant that came from someone else, or they have an autoimmune disease like lupus or one of the other diseases, or rheumatoid arthritis, where we try to tamp down activity of B cells and T cells, especially because they tend to drive those diseases. When we suppress the immune response, then we can put people at risk for a variety of infections. There's other kinds of immune compromise. So when we talk about this, I think it's a little dangerous to just talk about it as if it's all on one scale, where there's sort of an up and a down. It's a complex topic. And I think one of the things that COVID is going to teach us is some of the nuances around immunocompromise, because one of the perplexing things is what Annie talked about, that initially people have this infection, they have fever, they may have cough, some other symptoms, headache, but they don't have so much respiratory problems. And then in the second week, the whole disease turns a curve. And we think that that's being driven by the immune system. And we think it could be overactivity of one part of the immune system and maybe underactivity of another. And the underactivity of the other could be due to the virus messing with our immune system. It has some genes that can change the way we respond to certain stimuli. We don't understand these things altogether, but we know that people who've had an organ transplant, who have HIV infection with depleted immune system because they have AIDS or at least advanced HIV infection, people who are on medicines to suppress their immune responses due to autoimmune disease, those people are at increased risk for the disease, but probably not all in the same way.
And so in terms of what to do, this is, we find that people within their homes or with people who they're constantly in contact with kind of let their guard down in terms of the infection control and social distancing. So as Dr. Ray mentioned, there's levels of immunocompromise. And those are, for folks who do have immunocompromise, this is a really good time to talk to your doctor about the steps that you need to take, because we can't quite calibrate our answers to what that individual person needs. But I would say in general, what you're looking at is making sure that you're really diligent about those infection control steps inside your home, dorm, whatever the case may be, and also thinking about the social distancing techniques that you use inside your home or dorm or whatever. We don't tend to keep 6 feet from those we're living with constantly, but those might be steps that are necessary, potentially masks. But like I said, these are general kind of ideas. Definitely need to talk to your doctor about what steps are needed in this instance.
Another question was, how do masks help you? And as some people aren't still wearing masks in public, and is there a correct way to wear a face mask?
I was tempted to talk about this earlier, so I want to at least volunteer to start some of this. There was some early mixed messages about masks, and I think a lot of that is based on experience with people who are not healthcare professionals wearing masks. We know that people who are not trained healthcare professionals, who have not been specifically trained in how to wear a mask, often wear it around their chin, below their nose, up on their forehead, and they mess with it a lot. And the problem is that when you're wearing a mask in public, or when you're wearing a mask, if you touch the mask, you now have contaminated your fingers with whatever was on the mask. And so and I've actually seen people in public pull their mask down to sneeze. Now, the whole point of wearing the mask is to catch that sneeze. So because the mask doesn't protect you from others so much, because the particle sizes that are floating in the air, if there are any from somebody who's 4 or 5 feet away, they're going to be small enough to pass through the mask. But the droplets that you make that are just leaving your nose or mouth are big because they dry out as they travel. So if you get the big loogies, the big globs of snot and cough and spit that come out of your mouth and nose, those will get trapped in the mask, and it's nasty, but at least it's not spreading. And you also cut down the velocity of the jet that leaves your mouth or nose when you cough, sneeze, or just speak. And so you'll help protect people around you if you wear that mask, but it means cinching it down so that you're actually breathing through the mask and that you don't touch it and get the stuff on your hands and then touch doorknobs and other things in your environment. So I think if you're going to wear a mask, you need to take it seriously and say, I put it on and I keep it on like a healthcare worker does. When you take it off carefully and think about washing it if you can. So a bandana is nice because, that's what I wear in public when I'm out doing my things, because a bandana I can wash and I can just wear it until I'm done with it. I don't wear hospital masks out in public because I feel like I'm being irresponsible, because I would say that those need to be saved right now for people who need them most. And also a bandana does most of what I need. It'll catch my snot and then I can wash it when I get home. These N95 masks that Annie and I wear when we're seeing people with COVID, they have a different function. They catch 95% of particles that are more than 3 microns in size. So that means that they catch large globules when we're up against a patient listening to their heart and lungs, and they cough or breathe. We actually have a chance of stopping some of those particles before we breathe them in. And so because we can't do the physical distancing when we're taking care of a patient, we need a mask that can block those particles when they're still big enough for that mask to stop. The face shield we wear helps too because it stops the jet from hitting us directly. So we wear an N95 and a face shield when we're taking care of patients on the wards. Those masks help us, but you should be doing, I say that distance is like a mask. If you keep your 6 feet of distance, it's like wearing the N95 mask. And if you wear that cloth mask, then you're also protecting others in case you happen to get close when you cough or sneeze.
And I'll just add that the masks are going to be a key feature of what we're going to be doing in public going forward. That's going to be, it's going to look, there are a lot of things that are going to look different, but the masks are going to be one of those things that are here to stay. And as Dr. Ray mentioned, this is one of those classic things. The masks that you're wearing, those cloth masks out in public, are more to protect others from you. And because what we are finding is people who are asymptomatic and have it can spread it. And that's really the core purpose of those masks out in public.
And that asymptomatic spread is the difference between this virus and the SARS virus from 2002, 2003. As I said, there were no transmissions of people who are asymptomatic to others in that epidemic, whereas asymptomatic spread is the thing that caught us by surprise on this epidemic.
Mhm.
Everyone should assume that you are infectious, and that's why you should wear a mask when you're in public.
Right. I've seen a lot of women putting any kind of mask or a band or some kind of facial covering, and then they touch their hair with their gloves on. It's like, oh, please don't do that. Just stop touching your hair. It's like they just now contaminated their hair, and then they're, because they didn't pull it back, and they just think if you just wear a mask that will do it, but it doesn't. And so it's hard for people without a background in healthcare to all of a sudden get a crash course in infection control.
And if I may, you mentioned gloves, and I love this topic. I don't wear gloves in public.
No,
I don't think they provide any protection, and people find that surprising, but it's important to know that this virus does not pass through your skin. So we wear gloves in the hospital. And again, people say, well, why do doctors wear gloves in the hospital? Well, the answer is I go into a patient's room, I put gloves on. I go in and I do what I'm going to do. I come out, I take them off. So that ritual of going in and considering my hands dirty the whole time I'm in the room, and on my way out, taking them off, and I Purell before I take them off, because when you pull the gloves off, you can also throw virus into the air if it's on the gloves. So if you just wear gloves out in public and you do all your stuff with those, then
Yeah.
you're not doing
Yeah, just-
anything
Yeah.
with those gloves except making yourself have a false sense of security. So I don't recommend gloves worn in public for the purpose of preventing the spread of this virus, because I don't think they serve the purpose that they serve in healthcare settings.
Right.
Another question, a question for I guess everyone is, what do you guys think would be a big change in, I guess, our society after post COVID-19? Like, what kind of, I guess, rules would change and all that?
I think people have been lax about covering their sneezes and coughs. I hope that that'll change a little. There's evidence that tuberculosis incidence fell in the United States before we had treatment for TB, simply by changing how people behave around spitting in public, some other measures like that, and also contact tracing and things. So some basic public health measures make a big difference even without all the testing and all the medicines. So I think that some of the basic habits we have around coughs, sneezes, maybe cleaning hands, maybe people will be a little bit better about washing. The thing that gives me some trepidation about this is we know that in San Francisco, the behaviors of people who are at risk for HIV infection really became much more careful for a while, until a new generation of people entered the community without a memory of the deaths that had happened from AIDS. And so I think we're going to have to be intentional about remembering the lessons from this epidemic if we're going to have changes in behavior that will help us manage the next one.
Yeah, I'd say on the meta level, like in terms of our US government, we prioritize protecting our country against armed invasion from other countries. And we haven't done a good job of preparing for pandemics or preparing for the consequences of climate change. I think what a lot of people are seeing is that what we're seeing is a worldwide threat that actually working together, country to country, state to state, we can overcome if we work together well and smartly. And I hope that I hope we can overcome coronavirus, and I hope we can live in a safer world in another year. But I hope we take these lessons in terms of taking science seriously, working together as a world to seriously combat worldwide threats, because I think climate change is our next one. It's a little bit slower moving, but it's going to be even more devastating than coronavirus.
I also wonder how many people were overextending credit cards before this epidemic,
Ooh,
and may
yeah.
have learned some lessons about overextending credit cards when you suddenly have loss of income. If you've overextended yourself and you're just counting on all those paychecks in the future, that's not a way to live. I will say, I grew up poor and had to pay my way through med school, had tremendous debt when I finished med school, and my wife and I lived very inexpensively, didn't go out to dinner or go to movies for a decade to pay off that monstrous debt, which was earning 11% of interest, by the way, at med school. So I think we learned never to carry a credit card bill from month to month. We've always paid our credit cards off completely every month, because we learned not to trust that things would always be good in the future. Maybe people will be more cautious financially going forward and separate wants from needs a bit better.
Like people and corporations and governments all together.
Right. I think so. I think there might be hopefully I'm hoping for this in the good in people, but what I'm finding is that people are kind of stepping up and being kinder to one another. That they, neighbors are looking out for other neighbors. I've had a couple, I call them the lawnmower fairies, show up, and all of a sudden my lawn has been mowed. So I was like, sweet, this is great. So it must be because everybody's home, and so people looking out for one another, connecting with one another, even if it's through Zoom or through phone calls and stuff. I think that's that hopefully that can kind of, we kind of got into this very materialistic, very, you know, just me kind of society. And maybe this is like broadening that we're just one person, but we have other people, there's families involved, and that maybe there's, you know, we kind of keep that in mind. But also I like what you were saying, Dr. Ray, about, you know, for the saving up your money and stuff, because that's something that the depression babies learned, because they lived through it. And then, you know, we got very prosperous, and people stopped doing that, and they're living for today, and they're not thinking about tomorrow. And they said something about the average person in their 50s has, or 60 in their 60s has less than $30,000 in the bank, and that the average person could not pay a $400 bill without using a credit card. They have less than $400. And so I don't know what they're doing now. So, but that was shocking that, you know, that you can't even make up $400. So hopefully that's a wake up call for our society.
And I think the other piece that I think about is, you know, when we talk about public health, it's that interconnectedness of people, and that our actions impact others as well.
Right.
And we're seeing that now with the transmission that's happening. You know, I mentioned how we treat people who are poor in terms of access to healthcare, in terms of the social determinants of health that we talk about. And I was just looking at some of the experiences in Singapore where they didn't pay as much attention to the laborers as a reservoir of disease. And so all the great things they thought they were doing, they ignored the poor people. And now they're having trouble controlling it. And we're going to see the same thing here, because honestly, that's how we as a society treat people who are poor. And it's going to hit up against some competing values here. If we really want to control it, we're going to have to treat everybody equitably during this crisis.
Another question was, do people who get coronavirus twice get slightly different types of it? And another one was, is it possible that the test kits are yielding false positives if the person who was tested had another cold, even if it's not the coronavirus?
I'll give a brief answer. I don't think we have a confirmed case of repeated infection with this virus. We have examples of people who tested positive and then a couple of negatives, then a positive, but we don't know whether those were false negative tests in between. There's no evidence yet of a repeated infection with this virus that I know of.
I haven't seen a case report of a repeated infection yet.
I'll just hope that it's going to be mild the second time.
Right.
I think we all hope that this might become a cold virus like the ones we've had before, but we can't count on that. And we still have about 95% of the US population that has probably not been infected. The upper bound estimate for the prevalence of antibodies in the Santa Clara study, even though it was seen as having too many false positives, even there they estimated 4% at the upper bound had been infected. So that means that 96% of people in that county, where they probably had a noisy test that was giving too many positives, 96% did not have those antibodies. So I think we can, to get herd protection, we need about 70 to 90% infected people who are immune. So we don't want to go down the route of depending on herd protection for this infection. We need to.
Yeah, it's too dangerous. So who's going to volunteer for that?
Can I say a little bit about treatment and therapeutics? We've talked a lot. So we don't have any known proven therapeutics that work. I think some of the first signals from the one of the largest United States studies should be coming by the end of May. That's for an antiviral called remdesivir, and the trial is run by the NIH. The maker of that drug also has 2 other trials in the United States, so the results from that might come before the NIH trial or after the NIH trial, but that's the remdesivir. That's a direct acting antiviral that in cell culture and in animal models of similar viruses like MERS and SARS appears to work. that those medicines typically work well if given very early on after symptoms start. So we'll have our first signal as to whether remdesivir is going to work. And then there are many other therapeutics that are in clinical trials now, and then drugs that are in development to then go into clinical trials soon after that. Also, people are working hard to find monoclonal, or to identify monoclonal antibodies that can be given as infusion to people. We think some of the first clinical trials of that will start in the summer. And right now there's open trials and kind of extended use of convalescent plasma. So if somebody has had SARS-CoV-2 and has neutralizing antibody titers, then in certain cases directed by the physician in the hospital, will be given to people either in the outpatient setting or with severe disease in the inpatient setting. So there's multiple different avenues that people are looking at. And actually the last thing I'll mention, so we're looking at direct acting antivirals, and we're looking at convalescent plasma and monoclonal antibodies. And then we're also looking at anti-inflammatory drugs because, as Dr. Ray mentioned, the inflammatory lung disease is typically what is killing people. And so if we can use some of our anti-inflammatory medicines in a smart way, then perhaps they can tone down that respiratory aspect of the severe disease. So these are all things that over the next 6 months, we'll hear a lot more about. And so I'm hopeful. I'm always hopeful. I'm hopeful that there will be a therapeutic that works in randomized clinical trials, hopefully in the next 6 months, but we will see.
Okay, so it's about time to wrap up. So I was going to see if any of you had any final comments, or if you want to say anything before we end this live stream.
Yeah, I'd just like to, my final comments are really around how each of us has a responsibility in this, that each of our actions add up to help slow the spread of this. And we were talking earlier about how, while we can certainly see severe disease in any age group, it does disproportionately affect older individuals, those with chronic diseases. And so even if you are healthy and doing well, younger, all those kind of good prognostic factors, you're still the keeper of other people's health, including those you care about. And so everything you do matters. So just remember that in terms of a public health approach.
Okay, that's about it. So if you guys have any other questions, if anyone has any questions, what resource would be the best place to contact?
So I think always your primary care doctor is the great first step to go to. The CDC and the World Health Organization have fabulous sites. The NIH has an amazing site. Our own Maryland State Department of Health has an amazing coronavirus site with links to all the resources specific to Maryland. But if you have specific questions about your own health, I think talking to your primary care doctor is the best way to go.
Your local health department.
And if you do have questions about COVID, you can certainly call the Anne Arundel County Health Department, 410-222-7256, and we can certainly answer your questions and or steer you in the right direction as needed.
Right. If anybody is interested in a career in lab medicine, you can call Anne Arundel. Well, you can, let's say, I'll just give you my email, ljdousette@aacc.edu. I'd be happy to start you on your career in the laboratory profession.
And thank you everyone for joining us in the live stream. And that's it for our live stream. Thank you.
Thank you, Ryan.
Great, thank you. Thank you.